Preparation Model Paper 13 on ASRB Plant Pathology. I manage Shankar Reddy, PhD Plant Pathology. So, in continuation to the previous one, that is 35, that is just a continuation. So, the shape of primary juice pores in saprolignia. So, generally, this saprolignia fungi produces two kinds of juice pores, that is called primary juice pores and secondary juice pores. So, this phenomenon is generally called as dimorphism, right? So which is having two kinds of juice pores, primary juice pores and secondary juice pores. They're asking just the shape. So generally this saprolignia that comes under this Oomycota group of fungi, the primary juice pore, generally the shape of primary juice pore is pear shaped. When it comes to the secondary juice pore, which is a kidney shaped. So 201, the shape of primary juice pore is a pear shaped, and the shape of secondary juice pore is a kidney shaped. So the phenomenon where it is producing the two kinds of juice pores is known as dimorphism or diplanetism, also we can call it. So this saprolignia cell wall is made up of chitin. Generally the Oomycota group of cell wall is made up of cellulose, but when it comes to a couple of fungi, even though they comes under Oomycota group of fungi like Achaila, Aphadogaila, saprolignia and Leptomitis, their cell wall is made up of chitin only. The next one, cucurbit yellow vein disease is transmitted by, they're asking how it is actually transmitted. So generally they transmitted by a fungi called Serratia marcens. So it is a bacteria, Serratia marcens. The next one is cucurbit yellow vine disease is sorry, just a minute, caused by, they're asking. So caused by Serratia marcens, transmitted by a squash bugs. Okay, just make a correction of this. Cucurbit's yellow vine disease is actually transmitted by squash bug caused by Serratia marcens. Okay, the next one is a sporangial proliferation is a characteristic feature of. So generally the sporangial proliferation also can be observed in fungi. This is a saprolignia. So there are a couple of special features this saprolignia having. This diplanetism or dimorphism, that is having producing two kinds of juice pores, primary juice pores and secondary juice pores. Again this sporangial proliferation also can be observed in the same saprolignia. For further more information, we have published in detail about this saproligniales and saprolignia in mycology section. You guys may refer to have much more information on this saprolignia. And the next one is dapple apple disease is caused by, dapple apple is a disease which is caused by, they're asking, generally it is caused by a viroid. What is the first sex hormone to be discovered, right? So the anthradiol, so anthradiol, ANTHERADIOL, anthradiol is the first sex hormone. So the fungi that can produce sex hormones was actually first initiated by Anton de Barry. The later couple of other scientists like John Draper and some other scientists started working on it. So the first sex hormone to be discovered is anthradiol. The second one is cryoprotection agents or cryptoprotective agents used for a long-term preservation. So generally for long-term preservation, glycerol 10 to 15% can be recommended, and one more is DMSO, which means dimethyl sulfoxide, that is also can be used. So glycerol can be recommended for 10 to 15%. That is one a long-term preservative, and one more thing is DMSO, dimethyl sulfoxide, that is also one more cryopreservative. The next one is who initiated etiology work in India. So generally, sorry, aerobiology, not etiology. So aerobiology work was in India was first started by E.D.D. Sinningham or Cunningham, we can call it. ORF rings is present in Badna virus. So ORF in the sense open reading frame. So open reading frame, generally the virus contains open reading frames. So how many number of open reading frames? Each and every virus have different kinds of open reading frames. Generally you can take tobacco mosaic where it contains 4. So like that each and every virus has their own number of open reading frames. Generally this Badna virus contains 3 open reading frames. The next one is the tobacco mosaic virus. Just now I told you the tobacco mosaic virus contains 4 open reading frames, whereas the open reading frames present in is RTV. RTV also contains 4 open reading frames. The next one is the cassava bean mosaic virus. So cassava bean mosaic virus also contains 4 open reading frames. And the next one is open reading frames present in soybean chlorotic mottle virus. Soybean chlorotic mottle virus also contains 4 open reading frames. Then Curto virus contains 5 open reading frames. Then cauliflower mosaic virus contains 8 open reading frames. And then citrus virus contains 12 open reading frames. So TMV, RTV cassava bean mosaic virus, soybean chlorotic mottle virus contains 4 open reading frames. Curto virus contains 5, cauliflower mosaic virus contains 8, citrus tristiza virus contains 12 open reading frames. The next one is so Badna virus. The virus comes under the this Badna virus. Just they are asking a couple of examples. So what are all examples of virus that comes under this Badna virus group? Like cocoa swollen soot virus, which is transmitted by mealybugs, banana streak virus. So these are all the couple of examples that comes under this Badna virus. And indicator plant for chrysanthemum stunt viride. So for every viride and virus also indicator plant will be there. So the common indicator plant for virus is goose foot, Chenopodium amaranticolor, I think. So generally every virus and virides have their own indicator plants. So they're asking the chrysanthemum stunt viride having the indicator plant. The chrysanthemum stunt viride having the indicator plant, chrysanthemum morifolium. Chrysanthemum morifolium is the indicator plant. And coconut kadang viride indicator plant is Cocos nucifera, that is coconut. So citrus exocartosis is a trifoliate orange. So this indicator plant is important, trifoliate orange. So potato spindle tuber viride indicator plant is generally tomato can be recommended. Tomato and amoeboid inclusion bodies are produced by. So every a virus produces inclusion bodies as a identification factor. For example, if you see tobacco mosaic virus produces X bodies. So bullet shaped inclusion bodies or granular inclusion bodies, such kind of inclusion bodies we might have seen in the virology section itself, right? So every virus produces their own inclusion bodies. So they're asking amoeboid kind of inclusion bodies are produced by virus. So turnip mosaic virus produces amoeboid kind of inclusion bodies. Crystalline inclusion bodies. The next question is fully crystalline inclusion bodies are produced by. So alpha mosaic virus, and then one more example was there. I couldn't able to recall, but alpha mosaic virus is one example. And angular leaf spot of cucumber is caused by this angular leaf spot of cucumber. It's a very important bacterial disease which is caused by Pseudomonas syringae pathovar lacrimens. Pseudomonas syringae pathovar lacrimens, which causes angular leaf spot of cucumber. So endospore decolorizing agent. So what is the decolorizing agent used for endospore? So generally commonly water can be recommended for decolorizing this endospore. So mordant used in endospore staining. What is the mordant used in endospore staining? So heat, generally heating can be recommended. Other things also will be there, but this is the very important one, endospore staining agent. So what is the staining agent used in endospore? There is a staining agent called malachite green. So malachite green generally recommended for the staining of endospore. The next one is soybean rust is caused by. There are two types of rust: Asian soybean rust and normal soybean rust. Pacospora pacarizae and Pacospora myobomia, which causes Asian soybean rust and general soybean rust. So you guys can remember two things. Soybean rust is caused by Pacospora pacarizae and Pacospora myobomia. Myobomia probably Asian soybean rust, I think. General soybean rust, old soybean rust is caused by Pacospora pacarizae, I think. Probably, I'm not sure. But this generally this rust is caused by Pacospora pacarizae and Pacospora myobomia. You just remember two things. And grapes rust is caused by grapes. We may studied this downy mildew and powdery mildew, but grapes also rust disease was there, which is caused by Pacospora uvitis. Pacospora uvitis, which causes the rust in grapes. In bacterial cell wall, that is peptidoglycan, two sugar molecules are connected by. So what are all the two sugar molecules? For example, NAG and NAM are the two sugar molecules, right? N-acetyl glucosamine and N-acetyl muramic acid constitute to form beta 1,4 linkage. So in this two sugars, which are connected by, so what is the bond? They are asking glycosidic bond. The bond is a glycosidic bond. Siderophore selective medium, there are CAS medium can be told, like chrome azurol S, there is a selective medium. Phenolic theory, where it explains the red onions are resistant to onion smudge Coltotrichum circinens when compared to white onions due to the presence of phenolic compounds catechol and protocatechol as given by. So simply they are asking phenolic theory was discovered by. Phenolic theory was discovered by J.C. Walker in around probably 1940, I think. So he explained a theory called phenolic theory, which means Red onions are resistant to onion smudge disease. Onion smudge is caused by Colostotrichum circinus. Sometimes they may ask phenolic theory is related to which disease? Onion smudge. Onion smudge. Sometimes they may ask pathogen also, Colostotrichum circinus. Okay, red onions are resistant to onion smudge because the red onion contains two phenolic compounds. The phenolic compounds like catechol and protocatechol, they act as a like a toxins against this pathogen. So due to the presence of this catechol and protocatechol in the red onions, they are highly resistant to onion smudge disease. So the theory was given by J.C. Walker. The next one is lead blight of potato was first introduced in India. So it is believed that lead blight of potato was first introduced in Nilagiri Hills. Nilagiri Hills. The next one is sugarcane ratoon stunting is a. So generally this sugarcane ratoon stunting is caused by Lifesonia xylem subspecies xylem. Before it is called as Clavibacter xylem subspecies xylem. So it is actually a xylem inhabiting a gram-positive bacteria. Xylem inhabiting gram-positive bacteria. This xylella. A Lifesonia xylem subspecies xylem. Sugarcane ratoon stunting is a we can write pathogen as well as we can also write xylem inhabiting gram-positive bacteria, something like that. And the next one is the bacterial speck of tomato. It is a bacterial disease which is caused by Pseudomonas syringae pathovar tomato. Bacterial spot of tomato, bacterial speck of tomato is caused by Pseudomonas syringae pathovar tomato. But bacterial speck is caused by, it is also a bacterial disease which is caused by Xanthomonas vesicatoria. Then next one, bacterial canker of tomato. This is also important. Three are bacterial diseases only. Bacterial canker of tomato is caused by Clavibacter michiganensis, subspecies michiganensis, enters into the plant cells through trichomes. Such kind of things might be studied in bacteriology through natural openings or entries, like a mode of entry of plant pathogen. Probably this pathogen enters into the plant cells through trichomes, I think, probably. So I repeat once again, bacterial speck of tomato is caused by Pseudomonas syringae pathovar tomato. Bacterial spot of tomato is caused by Xanthomonas vesicatoria. Bacterial canker of tomato is caused by Clavibacter michiganensis, subspecies michiganensis. The next one is 239. Bacterial wilt of tomato is caused by, it is also a bacterial disease which is caused by Ralstonia solanacearum. Fixing solution in ELISA. What is the fixing solution used in ELISA? Sodium hydroxide NaOH generally can be used. So block buffer used in ELISA. So BSA like bovine serum albumin is generally used as a block buffer. I repeat, fixing solution is the sodium hydroxide and block buffer is used is bovine serum albumin. So black rot of sweet potato is caused by black rot of sweet potato is caused by pathogen Ceratocystis fimbriae. In disease of sweet potato we might have studied this. Black rot of sweet potato Ceratocystis fimbriae or fimbriata sometimes. The next one is a black rot of grapes is caused by. So black rot of grapes is caused by Guignordia budweli or bidweli. So black rot of cabbage is caused by Xanthomonas campestis pathovar campestis, right? Xanthomonas campestis pathovar campestis. So black rot of tea is caused by Corticium thiae. Black rot of coffee is caused by Corticium salmonicolor. Black rot of vanilla is caused by generally black rot of vanilla Phytophthora species, probably Phytophthora palmivora, some kind of thing. Phytophthora species, I think. So the next one is the banana shaped basidiospore is produced by. So generally this Exobasidium vexans generally produces this banana shaped basidiospore. Exobasidium vexans in the sense disease of coffee, right? Exobasidium vexans. Probably disease of tea or coffee we can see this. I couldn't able to recall it, but Exobasidium vexans generally cause disease in tea or probably in coffee. Then what are the gases recommended for gas sterilization? Generally formaldehyde, ethylene oxide, propylene oxide generally can be recommended. Virides are classified based on actually. So based on CCR, that is a central conserved region is the prime base for this classification of virides. CCR means a central conserved region. These virides are actually two groups, right? Avicenna viride, Pasp viride, both are classified based on this CCR, central conserved region. So medium valuable in differentiating a virulent colonies of a bacterium from avirulent colonies. So generally the tetrazolium chloride agar medium can be recommended for this differentiation of bacteria from virulent to avirulent. So chemical used in negative staining of ISEM, immunosorbent electron microscopy, that is uranyl acetate, can be recommended. Uranyl acetate. Which microscope is regarded as a student microscope? Like a bright field microscope, right? So the bright field microscope generally considered as a student microscope, and if you want to buy, it will be suggested to go with this bright field kind of microscope, so that that will be very useful for students since at student level. So slipper shaped sporangia, might we have studied this slipper shaped conidia and all those things, right? Slipper shaped, it is supposed to be not sporangia, it is supposed to be slipper shaped conidia, I think, probably. Various kinds of pathogens produces, right? So just keep this question away from it. I can't able to answer it right now because slipper shaped conidia, I think, like Physoderma, such kind of things, Physoderma geomedis or Physoderma medis kind of things will be produced. Sporangia, no, there is no such kind of sporangias are there, right? Only maybe globos, such kind of things will be there, but the slipper shaped sporangia. Anyway, I will recall it and I will let you know in the further editions, or else I might be commented in the comment section. Powdery mildew of wheat is caused by, so wheat powdery mildew is caused by Erysipec graminis. Now it is changed to Plumeria graminis. Not only wheat, powdery mildew of barley is also caused by same, that is a Plumeria graminis or Erysipec graminis. The next one is cholesterol present in Oomycota. So Oomycota cholesterol is fucosterol, whereas true fungi contains cholesterol is ergosterol. Which disease is considered as the jaundice of wheat? So stripe rust, that is caused by Fucsinia striformis. This stripe rust is also known as a yellow rust, which is generally considered as a jaundice of wheat. Because of that yellow color discoloration, all the stuff, we can consider it as the jaundice of wheat. Presence of pineapple odor in sugarcane citrate. So sugarcane citrate, generally this this pineapple color smell can be appeared, right? So what is the reason exactly? So generally, if you see, kernel bunt infected plants produces foul smell due to trimethylamine. Here, due to sugarcane citrate infected plants produces ethyl methyl acetate. I repeat, ethyl methyl acetate. Due to the presence of ethyl methyl acetate, this sugarcane produces citrate, I mean pineapple odor. And the next question is the kernel bunt. Kernel bunt infected plants produces foul smell due to the presence of an acid called trimethylamine. But this sugarcane citrate is ethyl methyl acetate. The next one is red gram sterility mosaic virus is transmitted by. So red gram sterility mosaic virus is transmitted by Eriophyid mate, Asaria kajani, right? In a semi-persistent manner. The answer is Asaria kajani, Eriophyid mate. Okay. The next one is the largest known viroid. So the smallest viroid is a coconut kadan viroid, which contains 246 nucleotides. The largest viroid is a chrysanthemum chlorotic motile viroid, which contains around 399 to 401 nucleotides. So autogamous genus of Ascomycetes, they are asking. So autogamous genus of Ascomycetes probably Sparrowtheca, I think, I'm not sure, probably. So which nematode produces a giant cell, like 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 cell kind of things? I think this might have, we have studied this root knot. The name itself indicates knot. Knot means some kind of what can I say? Some kind of bulger kind of structures, or we can say giant gall like structure or cell like structures are produced by root knot nematode only. So vitamin deficient in Phytophthora. So in Phytophthora generally biotin is no, not in biotin. Yeast is biotin. So yeast is deficient in biotin, but this Phytophthora is deficient in B1, vitamin B1, I think. So naked hyphae producing genera. Naked hyphae produced by various kinds of genera like Bipolaris, Alternaria, Penicillium, Cladosporium. All these kinds of produces naked hyphae. So non-spore forming fungal genuses. So it produces no spores. You can take an example of resting spores like Sclerotium, Rhizoctonia, Claviceps. All this produces non-spore forming genera only. It produces no spore. Sclerotium, Rhizoctonia, they produces resting spore. That is Sclerotia, right? Epigram is a forecasting model that is developed for. So this model is developed for wheat powdery mildew. Sorry, barley powdery mildew, I think. So barley powdery mildew, may probably this is also recommended for wheat powdery mildew, I think, but epigram is generally developed for barley powdery mildew. Cumulative winter temperature index, that is the CWTI, is related to. So this cumulative ventral temperature index is generally related to Stewart-Wilthoff con. Why it is called as Stewart? It was first discovered by Stewart in 1895. Erwinia trachyphylla, right? Uh, the Stewart-Wilthoff con is caused by. So that transmits to two kinds of beetles will be there. Edinburgh beetle. No, no, not that. I think some other things will be actually forgot. But anyway, this cumulative entry index is related to Stewart-Wilthoff con. That I will the insect vectors and all those things I will try to put it in some other PPT. The Voronin bodies can be first observed in. So Voronin bodies are first observed in a fungi called Ascobolus. So enzyme act as a toxin. So which enzyme generally act as a toxin, right? So lecithinase, there is an enzyme called lecithinase which act as a toxin. That enzyme act as a toxin. The next one now, virus family that infect plants, animals, humans and all those things. So Rheoviridae and Rhabdoviridae both are the viruses that infect not only plants, animals, humans and as well as insects. Enzyme containing viruses. So which virus contains enzymes? Again same Rhabdoviridae and Rheoviruses which contains enzyme. Probably that might be the reason that can not only infect plant, these enzymes also facilitate to help these animals, humans and insects as well. And the next one is a pathogen containing both biotrophic and necrotrophic nature. Uh, this Puccinia, like Puccinia rigonita, especially we can say it contain both biotrophic and necrotrophic nature. Plant pathogen which produces telephone booth like symptoms. All viroids like coconut kadang kadang viroid, coconut lethal yellowing, and chestnut blight. All these plants produces all these diseases produces telephone pole like symptoms. I would say coconut kadang kadang viroid. And all majority of the viral disease produce the same kind of things only, but generally we would say coconut kadang kadang and coconut lethal yellowing, the typical symptom is the telephone booth like symptom, because the infected plants bears no leaves, no branches, nothing. It exactly look like a telephone. I mean, so all the branches will fall down and exactly like a pole. That's why it's called as telephone. And chestnut blight infected plants also can look like this telephone booth like symptoms only. Anomorph of cordyceps. So cordyceps anomorph Toliposporium, probably I think Toliposporium something. Potato virus X is transmitted by, generally this potato virus X is transmitted by fungi, that Syncytium endobioticum. And potato mop top is also transmitted by this Syncytium endobioticum, I think. So dome shaped Aprosoria is produced by a rice blast, generally produces this dome shaped Aprosoria, which contains high amount of glycerol, help to penetrate inside the plant cells. Example of fungi which contains some melanin in this Aprosoria, again same rice blast, verticillium, cochleobolus, alternaria, all these pathogens contains melanin in this Aprosoria, but generally we based on the options we might choose the options, but this rice blast is highly famous, and this cochleobolus also generally bipolis. Okay, key virulent factor for Colrotrechum. So this Colrotrechum produces an enzyme called pectin lyase, so that act as a key virulent factor. Slipper shaped, again this slipper shaped sporangia is produced by. So I will refer this question once again. Probably I couldn't able to recall it what is exactly this slipper shaped sporangia. Anyway, I will recall it and I will definitely comment below in the comment section. Alternate host for sorghum and maize rust. So sorghum rust alternate host is Oxalis corniculatus. We can also call that as a sleeping beauty, right? So alternate host for bean rust. So bean rust alternate host is Trigonella polycerata. Trigonella polycerata. I think all are alternate host only. In the mycology section and epidemiology section, we might have done this alternate host for all the crops. You please go through to have and better to just recall it again, better information. There we have provided in detail about information about these alternatives. Anyway, sorghum host or maize host, Oxalis corniculatus, we can call it sleeping beauty. Alternate host for bean rust is Trigonella polycerata, and the alternate host or collateral host for sunflower necrosis is sunflower necrosis is generally Parthenium, I think. So then alternate host for collateral host for beans anthraconosis, again same Trigonella polycerata. Alternate host or collateral host for flax rust, this is important, Linum mysorens. Linum mysorens. The next one is alternate host or collateral host for cutted disease of cardam. It is a weed host, subabulatum or something it will come, probably Ammonium subabulatum, I think so. The next one is 287, bacterial blight of rice. This is Lersia hexandra, and panicum repens, various kinds of things will be there, but generally this Lersia hexandra is important one. And this is also alternate host for brown leaf spot and rice blast as well. Antibiotic bulbiformin is used to manage which disease? So bulbiformin generally red gram wilt. Probably yes, red gram wilt. This bulbiformin antibiotic is can be especially used against to manage this red gram wilt. Compound teleospore is produced by the genus. So compound teleospore phragmidium. We know very well this compound teleospore is produced by phragmidium or crust-like teleospore, right? So bicelled ascospore is produced by uh, bicelled ascospore is produced by this pathogen apple scab venturi anaqualis, I think. Yes, probably bicelled ascospore venturi anaqualis also look like a slipper, right? Sandalwood is an example of, so sandalwood kind of parasite they're asking. I see sandalwood is the best example of partial root parasite, and Rafflesia arnoldi, and it is also one of the largest flower producing a plant parasite, which is a total root parasite or complete root parasite. Example of unicellular fungi. So majority of the fungi are multicellular, but yeast is generally considered as unicellular. Fungal cells can be stained by lactophenol cotton blue. Lactophenol cotton blue can be generally recommended for staining. Lactophenol cotton blue. Phytoplasma is only grow in phloem because this phloem contains excessive amount of sterols. So these sterols are the very important compounds for the growth and to stay in the phloem vessels. Diluting agents, so there are many various kinds of diluting agents are there, like talc powder or clay minerals or various kinds of things generally can be recommended. So emulsifying agents, again compounds like carbohydrates, various kinds of organic amino acids, lipids can be used as emulsifying agents. Spreading agents like soap, mineral water, glycerin, terpenes can be recommended as a spreading agents. So diflocculant agent. So diflocculant agent like milk, plant gum, resins, gelatin kind of material can be used as a diflocculant agents. So which are morels? They're asking yellow morel. So in morels also there are a couple of kinds of morels are there. Generally, Marchella esculenta is considered as a yellow morel. The next one again same yellow morels, Marchella esculenta. And 301 black morels, Marchella elatia, and early morals, Verpa Bomi, I think. So I don't know why the probably yellow moral or black moral, maybe that indicates the correct. Why it is called early moral, I don't know. Maybe it grows much more faster compared to the other morals. So generally this early moral is Verpa Bomia, and coffee ring spot virus and citrus leprosis virus is transmitted by spider mites. This is a very important one. Coffee ring spot and citrus leprosis is considered transmitted by spider mites. The sentence rust never sleep. So that was actually given by, that was actually given by very famous man, and the only person in the agriculture sector that is a Nobel laureate. Norman E. Borlaug is considered as father of Green Revolution. He got 1970s Nobel Peace Prize because of a Green Revolution. So he is the one who first told rust never sleep, because we know how dangerous this rust diseases are. More than 300 races, still it's continuing to infect the crops. Even though we develop various kinds of resistant varieties, still it can manage to infect, right? So that's why I told rust never sleep, and is also one who initiated this Borlaug Rust Initiative. SIGAR consulted a group of international research institute, like various kinds of research international organizations that comes under this group for especially working on agriculture aspects. So, and he is the one who also initiated the CIMMET, Centro International de Metro Maize, Aroma Intivi Maize Trigo. Very simple, International Research Center for Wheat and Maize. Okay. So the rust never sleep was given by Norman E. Borlaug. So who discovered the sexual stage of Phytophthora? So Phytophthora sexual stage was first discovered by Clinton. So generally this asexual stages and all those things was discovered by Anton de Berry, we know very well. But Anton de Berry, he tried until his last breath to find this a sexual spore of Phytophthora infestans, so that we can say this is the center of origin of Phytophthora. And later in Clinton 1920s and 1930s, he identified the source of inoculum is from some parts of somewhere from Mexico. Four-cylinder bacterial filter was invented by Charles Chamberlain generally. Plant viruses couldn't multiply inside the insect vectors. I repeat, plant viruses couldn't multiply inside the insect vectors. So some viruses can multiply, but this concept was actually told. They're asking black. Okay. Carotene group of fungicides are especially recommended for, generally this carotene group of fungicides recommended for powdery mildew disease, right? So the general one, the next one is false smut of rice, false smut of false smut of rice was first reported by. So first reported in Kerala, I think it was reported by Cook et al. I think so. So Pocan boeing disease of sugarcane is caused by, so various species of Fusarium. Generally we used to consider it as the Fusarium moniliforme. So for further more information, students can refer my book, A Vision into Plant Pathology, a complete student version. Further doubts, clarification, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.